Enjoy the session.
Well, good afternoon, everyone. Don't be shy, you can come to the front. It's going to be much easier to listen to me, because I never use this properly. So welcome to this session of the data track. This afternoon we have three very interesting talks, one proceedings and two highlights. And I think with not too much delay, we can just introduce, well, I'll wait 2 seconds people to finish entering. In classes, always more people, more students entering after the time than in time. So we can go ahead. So as I said, welcome, and I will just introduce the first speakers, if I can get the slides. I think, sorry, the name, Gokce is the right pronunciation? Gotche Uldogan, that is going to talk about exploiting protein biochemical language models for targeted drug design. So, go ahead.
Hello, everyone. I'm Gökçe, and I'll talk about our work exploiting pre-trained biochemical language models for targeted drug design. And so, as you may know, or you may not, the chemical space is vast, and only drug-like compounds have been estimated to be on the order of 10 to the 60. Therefore, identifying a compound with affinity towards targets of interest is like finding a needle in a haystack. And achieving this goal with experimental validation is infeasible. Thus, computational tools have been developed over the years to address this issue. And de novo drug design is one of the computational approaches which allows navigating the chemical space efficiently by generating novel compounds from scratch. And recently, deep generative models have been applied in de novo drug design, and these models require either structural data or known active compounds towards the target of interest to be able to generate target-specific compounds. However, such information may not be available for some proteins, especially novel ones. So in this study, we approached the targeted drug design task from a language perspective and formulate this task as a translation from protein language to chemical language, thereby eliminating the need for structural data and known active compounds. And the motivation behind this idea is twofold. First of all, chemists' approach to target-specific drug design is similar to translation task. Given a target protein, chemists have idea about which compounds can have binding affinity towards it. So this task can be viewed as similar to translation from protein space to chemical space. Secondly, proteins and chemical compounds can be represented as sequences, can be represented with sequences like sentences in natural languages, so we can view them as domain-specific languages. And in addition, proteins and chemical compounds are also composed of functional units or groups, as sentences are made up of words in human languages, so we can adopt some segmentation methods from natural language to tokenize such sequence into units that are akin to words in natural languages. And here in this study, we represent proteins with their amino acid sequences and chemicals with SMILE strings, which is a text-based representation for chemicals. And then targeted drug design can be framed as a translation task from protein language, whose letters are amino acids and words are identified by tokenization algorithms, to chemical language, where atoms correspond to letters and words are identified by tokenization similarly, as seen below in this figure. And this approach can be combined with any machine translation model. And here in this study, we use transformer-based encoder-decoder model, which is the state-of-the-art architecture. And the model is composed of stacks of encoder and decoder blocks, where the encoder takes protein sequence as input and outputs a representation for this protein. And while the decoder takes smile string and produced protein representation and outputs next chemical unit probabilities. And the key feature of this model is the attention mechanism, which appears in each block and captures the dependencies within individual sequences and across sequences. So this model requires pairs of sentences for training to learn translation. And in this setting, these pairs correspond to interacting protein ligand pairs. However, this data is limited compared to unlabeled protein and chemical compound sequences. And the question is how we can utilize this vast amount of data. And one possible way to learn representations based on such data and then use such representations in our task, targeted drug design. And here, language models, which trains with the objective as predicting, assigning probabilities to sequence of words to learn representations, and such models have already been used recently to learn protein and chemical language models. And in this study, we propose strategies to exploit these models to initialize targeted drug design models, to enhance generalizability and boost performance of our models. So the models we have used here are protein Roberta and Camberta models, which are both encoder-only transformers and are trained with mask language model, where the aim is to predict mask tokens in the input. Note that these models focus on both the left context and the right context of the input. And we propose two strategies here, one-stage strategy and two-stage strategy. In one-stage strategy, we use protein Roberta weights to initialize decoder of the targeted molecular generation model, and similarly, the decoder of our model is initialized with Camberta weights directly. However, as I mentioned a couple of seconds ago, Camberta is a mask language model and focuses on both left and right context. However, molecule generation task requires a causal language model, where the aim is to predict next word rather than mask token, and should only focus on the left context. So here in this strategy, two-stage strategy, we also included another stage where Camberta is adapted to molecular generation task, so that in the second stage, the model can focus on only learning targeted drug design. And this adapted model, we call this adapted model Camberta LM, and this is used to initialize the decoder of the targeted drug design model in the second stage. And these targeted drug design models are trained with interacting protein ligand pairs to learn the translation. And to investigate the utility of our approach, proposed approach, we trained worm-started models, encoder-decoder base using one-state strategy, and encoder-decoder LM using two-state strategy, as well as a baseline T5 model, which is a transformer model trained from scratch. And we used compounds from Moses to train Camberta and obtain Camberta LM, and we also used filtered protein ligand pairs from binding TB to train our targeted drug design models. Once the models are trained, we can use decoding strategies to generate molecules, and here in this study we use sampling and beam search. And for the evaluation, we use benchmarking metrics from Moses to assess the validity, uniqueness, and novelty of molecules, as well as their similarity to known active compounds with several metrics, including Fresha-Chem net distance, scaffold similarity, and nearest neighbor Tanimoto similarity. We also performed docking to assess whether generated compounds have affinity towards the targets, and to achieve these, we docked a number of proteins against 3 sets of molecules. Random set corresponds to the sets randomly sampled from data set. Active set comprise the known active compounds for the particular target, and generated set consists of the molecules generated for the particular target. And this set yields a distribution, a docking distribution. And we would like to generate molecules that are distinguishable from the distribution of the random set and similar to active set, so that we could understand generated molecules have affinity towards the targets of interest. And so we first validated the performance of Camberta LM. We benchmarked it against the baselines from Moses benchmark. And here we see the results. The best scores are highlighted in bold, and the second best ones are underlined. And we can see that Camberta LM obtains the best or the second best scores across metrics, suggesting that it performs comparably to or better than the baseline models across metrics. Next, we evaluated our worm-started models, targeted drug design models with benchmarking metrics, and we saw that worm-started models outperforms the baseline transformer trained from scratch in terms of validity as well as similarity metrics. And we also, next we evaluated whether the generated compounds are active against targets of interest. To this end, we computed the number of proteins where generated molecules are significantly better than random ones and similar to active molecules. And here we see that worm-started models can generate active molecules towards target of interest for a higher number of proteins compared to the baseline T5 model. And the top performing model here is encoder-decoder base trained with one-state strategy, which can generate active molecules, molecules better than random ones for all of the proteins, 11 proteins. We also ask whether these generated molecules have, these generated molecules are specific to these targets, and we performed another docking to compare the molecule set generated for these particular targets with the molecules generated for the rest of the targets to see whether the molecules are specific or not. And we see that this model, the worm-started model with the one-state strategy, can design target-specific molecules for 8 proteins out of 11 ones. So, in short, we approach targeted drug design from a language perspective and frame this task as a translation from protein language to chemical language. and this allows us to generate molecules using only protein sequence information. And we investigate two warm start strategies to exploit the representations learned by pre-trained biochemical language models. And our experiments revealed three findings. First, warm started models outperform a baseline model trained from scratch. Second, warm started models can generate compounds with affinity towards targets of interest. And last, warm started model with the one-stage strategy can design target-specific molecules. So I would like to end my presentation with acknowledgements. I thank my mentors and co-authors, Arjun Özgür, Elif Özkırımlı, Kutlu Ülgen, and Nilgün Karalı, as well as our project members. And I also would like to thank our funders. And that's the end of my presentation. Thank you for listening.
Okay, thank you for the talk. We have time for some questions. Not too much.
Yeah.
Nothing from the chat.
We heard this morning keynote about interpretability.
Yes.
And my question, can you have a way to interrogate the model and ask maybe what are the amino acids or what are the parts of the molecule that are interacting?
That could be a future work for this study. Yeah, we have the attention mechanism we can explore and to see whether we can find some patterns there between amino acids and chemical units. Thank you for the question.
Or? A bit shy, a bit sleep after lunch.
Just a comment. The docking program does exactly that, what you're asking.
I was curious, at the end you show results for a small number of proteins. Does that mean that each protein is a different model, or you train and just check with those proteins? Sorry. Yeah, I mean, these proteins, we are using the same model to assess all the proteins? Because, sorry, perhaps I misunderstood.
This is just for the one-stage strategy.
I mean,
I'm not sure I get
sorry.
the question.
So when you train this thing, is for a single target?
Yes,
So each target has a different process.
yes.
In a way, could you then include more information? Because if it's a target-specific process, you may probably not. Yeah, you can pretend of binding DB and everything, that these are general databases. But presumably you have specific information that you don't want to use because you are, well, for translation.
We
But
can incorporate such, I don't know what information, but into network by incorporating it into the input parts. I don't know.
so there's a way.
Yeah.
The point is, I understand that you are testing if just this concept of translation from one language to other is good enough. But if you actually want a compound,
Yeah,
you will
sure.
put everything there.
For
So
sure.
it's a way to do it, I understand. So you can add
Yes.
that information.
There are some studies doing this for other tasks, like multi-objective optimization, but we haven't tried that
Okay.
yet. Thank you.
Any more questions there? So if not, thank you.
Thank you.
So my ability to remember names is terrible. So next talk is by Barbara Grabo. Thank you. Barbara Grabo from Brussels, scaling up oligogenic diseases research with change subject with OLIDA, the oligogenic diseases database. Go ahead.
Okay, thank you for the introduction. So yes, I'm Barbara. I'm a PhD student in the Inter-University Institute of Bioinformatics in Brussels. And today I wanted to present a database that we developed for the study of oligogenic diseases. So if you don't know what are oligogenic diseases, don't worry. I'll start by introducing why we care about them and what they are. So in Europe alone, 30 million people are affected by rare diseases. These diseases are usually very severe, and the large majority of them have been shown to have a genetic origin. And better understanding this genetic origin can help improve the diagnosis of the patients, but also help develop new treatments for this disease. However, the genetic cause remains unknown for the majority of patients due to different problems with phenotypic variability, genetic heterogeneity of diseases, as well as incomplete penetrance of mutations. So this problem is known as the missing heritability problem, and one way to try to solve it is to try to look for more complex genetic causes to diseases. Indeed, genetic diseases have been shown to follow continuum. So on the one hand, you have monogenic diseases, which are caused by one variant, rare variant with a very strong effect. And on the other hand, we have polygenic diseases, which we have heard about yesterday in the keynote talk, which can be approached by using polygenic risk scores. But in the middle, we also have oligogenic diseases, which are genetic diseases which are caused by the interaction of variants in a small number of genes. This has been shown to be the case in some known diseases, such as Bardet-Biedl syndrome and Hirschsprung's disease. And one hypothesis is that the missing heritability problem can be solved by the fact that many diseases are actually caused by such oligogenic causes, but they're still hard to detect. So one important step into better understanding oligogenic diseases was the creation of the Digenic Disease Database, DIDA, by people from my group in 2015. And so this database collected information on variant combinations in gene pairs that were shown to be responsible for genetic diseases, and so collected information on 258 combinations responsible for 54 diseases. So from this database, many machine learning models have been developed to predict first different types of digenic effect, and then also the pathogenicity of variant combination in gene pairs as well as gene pairs. And so together, this database as well as these methods have led to a lot of research in the area and a huge increase in the number of combinations reported to be linked to genetic diseases. So this highlighted two things. First, we need to improve the data collection on this type of genetic inheritance. And second, the DIDA database was not enough to cope with the continuous increase. So in order to tackle these issues, we developed a new database called OLIDA, the Oligogenic Disease Database, which had three main goals. So the first one was to include oligogenic combinations which involved variants in more than 2 genes, which were not collected in DIDA. The second one was also to include new types of variants, such as copy number variants. And the third one was to actually have a detailed curation protocol that could help that assigns confidence scores to the different combinations in the database based on the evidence that associates them to a disease. So the data in OLIDA was collected by manual curation again. So what we did was just to search in PubMed for articles published with the keywords digenic or oligogenic. Then each article was reviewed independently by 2 curators who read the article, extracted the information, and assigned scores based on the evidence that was presented in the article. Then the 2 curators discussed this data to assign final consensus manual scores. In a second step, we annotated this data with information from external biological database in order to first obtain the genomic coordinates of the variants to normalize the disease names, but also to add new information, such as whether the genes are known to interact, for example, based on protein-protein interaction data, whether the variants involved were already reported as being pathogenic or linked to a disease in ClinVar. And based on this information, we also assigned knowledge scores, so scores of the evidence that is supported by external data. Finally, in the last step, the manual scores and the knowledge scores are combined into final confidence levels that are associated with each combination in the database. So in order to score the combinations, we looked at two types of evidence. The first one is the genetic evidence, which for oligogenic disease should prove that the segregation of variants together is linked with the phenotype of interest. And so this genetic evidence can be obtained from familial studies, where we look at the proband's relative and look at whether healthy individuals don't show the combination, and if variants alone are responsible, can be responsible or not. Then this can also be obtained from cohort studies, where we compare, where authors compared large cohorts of healthy individuals compared to people with the disease. And we also scored the functional evidence that was associated with the combinations, which can be obtained from functional experiments, or also, as I mentioned earlier, the database of known protein-protein interaction data, etc. And so our premise for the curation protocol was that both genetic evidence and functional evidence was needed to show that a combination is responsible to disease. For each type of evidence, we assigned different levels. Based on detailed criteria that you can find in the paper. Just to give an example, for pedigree studies, we required for the strongest level of evidence to have information from both first-degree and second-degree relatives, and to show that, for example, for a digenic combination, each variant could be seen alone in a healthy individual, but each individual who carried both variants had the disease phenotype. So what we have in OLIDA now is that it represents a huge increase over the data that was collected in DIDA. So we now have data on 1,229 oligogenic combinations involved in 177 diseases. We now also collect data on copy number variants. So we have some combinations which contain about 62 copy number variants. And so, yeah, it's basically a fourfold increase compared to the original data database. And so in terms of confidence levels, so our scores were from 0, which represent absent evidence, to 3, which represents strong confidence. And so we can see that, unfortunately, based on our curation protocol, most of the combinations in OLIDA were assigned a score of 0. Even after the addition of external knowledge, which actually led to the improvement of substantial number. And so what does this show is that currently there are no standards in how to prove oligogenicity and how to prove that oligogenic variants are involved in an oligogenic disease. And so these standards are needed, and this is one of the main things that we wanted to show with this database as well, is that we need to find new ways to properly assess this, the involvement of oligogenic combinations in disease. And so we're also preparing a commentary on new standards that are needed, which is currently under revision. And so, yes, so the OLIDA database is available online. We made sure that it followed the FAIR principles on data management. So if you're interested in that type of data, all the different entities are organized in different tables with specific identifiers. It can be downloaded in different formats and is also available through a programming interface. We also made sure to annotate the data with external ontologies and databases. So for example, here for diseases, you can find the ORPHANET ID, the ICD category, as well as the OMIM references. And finally, all the curation protocol, the different criteria that we use to assign scores, as well as all the scores are actually available in the article and in the documentation of the database. And so, OLIDA has been published this year. I will just show you the reference to the paper, etc. At the end. But we already, in our group, started using actually the data to generate new applications. And so I just wanted to give two first examples of what we can do with this type of data. So the first one was done by my colleague Alexandra Ono, who presented a poster on it yesterday, and also a talk at the student symposium, if you had time to listen to this. Otherwise, you can also find it on the app. And so what he did is that he integrated this OLIDA data together with several databases of biological data in a knowledge graph, and on this trained fully interpretable methods that could predict gene pairs that are likely to be involved in an oligogenic disease. And this method can also provide explanations for what type of mechanisms underlie the potential pathogenicity of these combinations. The second applications I wanted to talk about was the development of a new variant combination pathogenicity predictor, so VARCOP 2.0. This one is presented at a poster session tonight by my other colleague Nassim. And so using only high-quality instances of OLIDA allowed to really improve the performance of the predictor compared to the first version that was developed based on DIDA. And also to reduce a lot the computational time, which will allow us to move towards oligogenicity prediction in whole exomes. And so, okay, to conclude, I have really three main messages that I wanted to give with this talk. So the first one was we've collected this largest repository of data on oligogenic diseases and the variant combinations involved in such diseases. And so feel free to use it. It's available online. You can try to check if the disease you're working with is known to have oligogenic inheritance associated with it, if the genes you're working with are known to be involved in such diseases, etc. Develop new machine learning methods as well, maybe. We need to have good data and good standards for the reporting of these variants, and so we've started to try to develop criteria based on our reading all this literature on them, but they're still open for more suggestions, etc. And so if you're interested and have ideas, feel free to come talk to me as well. And then, so I showed a few applications of what we can do with it, but there is a lot of research left. And so if you're interested a bit more in those applications, you can also go see my colleagues. And so in terms of perspectives, I had 2 points. So the first one is that manual curation is a huge amount of work. And if we want to keep updating this database frequently, we need to try to develop new methods. So we have also someone in our group working on a machine learning method, text mining method to be able to get this data automatically. And finally, we're aiming also to have this become a more community-driven effort for the database. So we already have a submission interface in the database. If you have new oligogenic data, you can submit it there. And if you're interested in participating, you can also email us at this address. And so finally, yes, so here is the article. If you're interested, it was published earlier this year in database. So it has all the detail of the curation protocol, etc. And yes, I want to thank everyone who made this possible, especially Charlotte and Sophia, who I worked with for the data curation, which was the biggest part of this work. And then OLIDA is available at this website over there if you're interested. And yes, thank to everyone else in my team and you for your attention.
Okay. Thank you for adjusting to the time perfectly, and a very nice talk. I don't see anything on the chat, but I'm not sure this is working because I tried. Just in any case, questions from the audience.
Hi. And with all of this amount of work that you put in, did you have time to look into the pairs or triplet of, genes that are involved? Are they usually paralogs? Are they on the same pathways? Are they overlapping? What biological insight did you get from this, the new set of oligenic?
Yeah, so there are like, so there are many, so we actually do give the different types of interactions that are known between the genes in the database, and so it's actually very complicated. There are many different, so either pathways, some are known to be directly interacting, some are paralogs, but just like this, I can't give you a direct answer of what are the ones that are the most represented. So yeah, that's why the knowledge graph approach is trying to find rules based on this. So if they're found often, yeah, in the same pathways, etc. But of course, it's the mechanisms are always different.
But nothing came out as a dominant mechanism?
Not that I can tell like this, no. Yeah.
Hello.
Yeah.
Thank you, Barbara, for the excellent talk.
Thanks.
I just have one question. You mentioned that we are able to submit our data, so you can integrate it in your database. Is there any criteria for accepting our, pair or anything that we are submitting? So what kind of criteria that you are evaluating and accepting this and integrating it in your database?
So the goal with this was to provide a repository of everything that was reported in the literature. So you just have to have it published somewhere. So we're just, the only really thing that we need is the reference of the publication. And then
So
we
it's
will
supposed to be published, so we...
Yes,
okay.
yes, yes.
Because my group are working in something similar, but it's not published yet.
Okay.
But our bio creators are able to find some associations between different oligogenic diseases. So just...
Yeah, so because we scored then based on all the evidence that was
I see.
that was obtained, so we need to have, a detail of what was, how it was proven to be linked to a disease, what's the proof of oligogenicity, the proof of, yes.
Thank you very much.
But otherwise, we include everything, we just assign different scores based on the different types of evidence.
So my question is, when you had all these diseases, but the confidence score was very low,
Yeah.
is it because these diseases these diseases that are low score, are they rather rare diseases or common diseases? Or
So
what kind of diseases are there?
the ones with the low scores?
Yes,
Or-
because most of them are low score, I think.
Yeah,
so
yeah, yeah.
anyway, so these diseases?
It's a combination of a lot of, most of the diseases we have in the database are rare diseases
Okay.
in general. And then there are some more common ones. From the top of my head like this, there is scoliosis, for example, which is a more common disease. I wouldn't be able to remember the scores that we attributed to this type of, but the large majority are rare diseases because, yeah.
Okay, thank you.
No problem.
So regarding the definitions of oligogenism, how do you put the limit of frontier in between oligogenism and modifier genes? Because if you have one gene creating a mild disease, others the same, and the combination is leading to a severe disease, it's a kind of oligogenism, but not exactly. So how did you manage that?
Yeah, so we try to collect this data as well. Also, whether it's known as a true digenic or true oligogenic or a gene with modifier. And so that's based on what the study, if the study is done, but so we include those modifier cases. And we try to label them if the evidence is enough that shows that, for example, yes, someone has more severe disease if he has more variants. And so, but often the evidence for that is never completely clear, as you're pointing out. So that's why it's also very important to try to find better methods to assess oligogenicity. But for me, I considered modifier cases like oligogenicity because it's defined as the way to better explain the phenotype is actually to have these extra variants. So the extra variants in the genes are responsible for explain the phenotype better. So it's an oligogenic case, but which we label usually a modifier effect.
Okay, thanks.
No problem.
Any more questions, the audience? If someone is online and couldn't put a question, apparently the system is back, so you can try again, if the case. I have a million questions because I do some of these things. But I never did more than one variant at a time. How bad the traditional methods work with these diseases?
How about, sorry?
The traditional methods, polyfem, PEMUT, you are not using for these hours, behave trying to predict this oligo.
The traditional, so how do people find these oligo? Yeah, so for familial studies, for example, they just look at how the segregation of variants is happening, and when it can't be explained by one variant alone, because you find an individual in the family who has the same variant and doesn't have the phenotype, that's how they search. So they usually search in gene panels, or now a bit with whole exome sequencing as well, and try to, yeah.
No, my question, sorry,
Okay,
if you use a traditional method to predict the pathology with these diseases, with these variants that are based in one variant each time, how bad is the result compared with yours?
Okay, so traditional methods for predicting pathogenicity is the machine learning predictors, for example?
Machine
Or?
learning
Okay.
predictors based on SNPs, based on...
Yeah, so we see that actually most of the variants that we have in OLIDA are predicted as pathogenic by those predictors. So we annotate them with CAD, for example, SIFT, polyphen, etc. And so the majority of them are predicted as pathogenic. Yes, I don't know if that answers.
So my second question is worth the work to do this if the traditional methods work? Sorry.
Yeah.
It's just provocative.
Well, the thing is, they work, but not for all of them. And then if the actual cause, like those methods also have a lot of false positives. And so if they detect these as pathogenic, but they detect in someone's data many pathogenic instances, how do you know which ones are the real cause? And so that's why we're also developing those new combination pathogenicity predictors.
Presumably in those cases that actually you have more than one variant that has a similar influence, perhaps your approach will be much better because none of the variants will be bad enough.
Yeah, also, yeah.
That's a good point to dig because, sorry, does anyone want to ask? I just keep doing it. No, sorry, because getting the data is terribly difficult. Not just for you to read the papers, for the people writing the papers.
Yeah.
Because if you start with a correlation 2, 3, 4 genes, it's impossible.
Yeah.
So
for like,
I would say we'll go to a polygenic gene score in 2 minutes if we went much farther than that. So for me, it's very important just to highlight what is different from the traditional approach.
Yeah.
That's important.
Okay. Thank you.
More questions? No more questions?
Sorry, I'm still there.
No more questions, you can go. Thank you very much. Okay, let's pass to Peter Smirnov, I think. Hope this pronunciation is correct. He's going to talk about PharmacoDB. Go ahead.
Yeah. Okay, hi everyone. As I was just introduced, my name is Peter Smirnov. I'm from the University of Toronto in Canada, quite a long way away. And today I wanted to talk about PharmacoDB 2.0, which is our latest release of a database of in vitro pharmacogenomic studies. Before I start, I should acknowledge my two co-first authors on this paper, Nick Tafezi and Shishira Nair. They weren't able to attend the conference because they were attending a different conference. And obviously this was the work of many people, so I should acknowledge all their contributions as well. So in the Habe-Keynes group, what we're interested in studying is finding predictive biomarkers for drug response in cancer. And in particular, we're interested in finding them from molecular omic technologies, looking for features that will differentiate the responding patients from non-responders. And one approach that we take to doing this is looking at cell line-based pharmacogenomic studies. So these were described in part by the keynote this morning, but the way that these data sets usually look is a large panel of cancer models, most often cancer cell lines because they're easier to work with, will undergo comprehensive molecular profiling together with a drug sensitivity screen. And the drug sensitivity screen, each cell line is treated with a panel of drugs. For each drug, there's a set of concentrations to form a dose-response curve. And then we'll summarize these dose-response curves into values summarizing the sensitivity, like IC50s or area above the curve values, or other metrics. And so the way we think about biomarker discovery is we're making an implicit assumption that the drug sensitivity is a function of the molecular state of the cell, and we're asking which of the molecular features are most correlating or most predictive of drug response. So fortunately for us, there's been a lot of data of this sort published over the last few decades. And this has come both from large academic projects as well as pharmaceutical companies sharing the screening that they do internally. And it's lucky to be in a data-rich field, but this also causes its own struggles. And when we started working with these data, what we struggled with was trying to integrate across different studies. So what we found is because there was no single repository for this sort of data, we would struggle when we tried to compare data sets. And right now, large projects such as those from the Broad and Sanger Institute, they have the resources to develop very comprehensive data portals that do data versioning well and integrate across the studies done at their institutes. Same thing for the NCI 60 from the National Cancer Institute. However, many smaller studies, they get published with the original publication, and then it's hard to integrate this data with the existing larger projects. And a lot of the work that we do is manual curation because you end up in situations where the identifiers used in these studies need to be manually disambiguated. My favorite example of that is there's two cell lines, KMH-2 and KM-H2. These are two completely different cell lines, but often researchers will try to remove special characters from strings, and you just end up with KMH2. So you have to go back to the data and try to disambiguate these sort of cases. And we found that doing the manual curation led to as much as 30% in overlap between two studies. So since we were doing this, we were putting in the effort to go through manually and figure out what all of these entities were. We decided to create a data resource to share this work with the community. And our goals were threefold. First, we wanted to help researchers discover the publicly available data. So to answer questions such as whether your compound of interest has any public data available, or for example, if you're interested in a particular cancer or tissue type, which data set would give you the most information for cell lines from that tissue. We wanted to help our experimental collaborators either identify the most sensitive or resistant cell lines that have been already screened, or look at what was the range of previous IC50s seen in an experiment so they could plan their own concentration ranges. And finally, we wanted to also, for computational biologists, provide an easy way to search through and identify what data exists. So if you want to train a machine learning model, you can find out what data is available for your drugs of interest. Or if you collect your own data and do analysis at your institute, you can find validation data sets for that. And so what we ended up doing was we went to the original study publications, to data repositories, we contacted the authors of these studies, and tried as much as possible to get the raw data for all the data that's been published. We pass it through a unified alignment, normalization procedures, curve fitting. We do quality control on the results that we generate, and then we map to unique identifiers. More on that on the next slide. And that goes into a database which we call PharmacoDB. On top of that, there's a web app at pharmacodb.ca. And if you want to know more about our data processing pipelines and the way that we make sure that all of this is fair, there's a talk on Orchestra, which is a data sharing and pipeline orchestration platform that we've developed in tomorrow morning's data session. So coming back to PharmacoDB, we've tried as much as possible to map to standard identifiers already used by researchers commonly. So for compounds, we map everything to PubChem because pretty much every molecule, almost every molecule is in there. We also map to Chembl, to drug target commons so that you can find target information, as well as to Reactome so you can see which pathways are being targeted by that compound. For cell lines, we leverage the excellent work done at the Swiss Bioinformatics Institute in developing Cellosaurus, an ontology of cell lines. And with their help, we map each cell line to the NCI Disorder's disease IDs. For genes, this was admittedly pretty easy. There's a lot of resources for this. We settled on Ensemble, and we allow users to search our database with gene symbols as well. And finally, for tissues, we leverage MSK's OncoTree, which is a classification of cancers into a tree, which ends at the top with a tissue based on the cell of origin for that cancer type. And so for our second release, we now have 10 of the largest data sets that have been published. In total, we've reprocessed over 6 million drug dose response curves in 1,700 cell lines and tens of thousands of compounds, a lot of them from the NCI-60. And I can't demo the web application for you, but I want to show you what it looks like. So if you were to go to pharmacodb.ca, you'll be treated to this homepage, and it prompts you to search for a study, a drug, a cell line, an entity that you might be interested in. So for example, if I'm interested in lapatinib, I will type that into the search bar, it auto-completes, takes me to a web page which summarizes the information we have on that drug. So you see all the data sets it's been tested in. You'll quickly see that it's FDA-approved compound. We have the structural information for this drug, as well as the links to other data resources. If I'm interested in identifying sensitive or resistant cell lines, I can click on the cell line tab. And here, across all of the 10 data sets that we've integrated, we identify the 30 most sensitive and most resistant cell lines, with the error bars showing variation both across biological replicates and studies. So in particular, here, one of the resistant cell lines is OVCAR5. If I'm interested in the data that actually supports these IC50 values that were calculated, I can click on that, and I get to see all the dose response curves across all of these studies on one plot. And together with all the calculated drug sensitivity statistics at the bottom. And whenever you find data on PharmacoDB, or a plot that you like, there's going to be buttons to download either the plot or the data behind that goes into plotting it. Switching gears a bit, for another example, if I'm interested in the drug dabrafenib, for those of you who work on cancer, you may know it as a BRAF inhibitor. We grab target information directly into our database, as well as if you're interested, for example, in differential response across tissue types, you can use the Tissues tab, and here we quickly identify that dabrafenib is particularly effective against skin cancer cell lines, which there's a lot of high prevalence of BRAF mutations in skin cancer, and other BRAF mutated cancers from other tissue types don't tend to respond as well to dabrafenib, and that's something you can quickly see in the database. So finally, if I'm interested again in looking at the data behind this, we have a tissue-centric view of all the dose response curves as well. So this helps you with two things: downloading the data for a particular tissue type, as well as you can individually go and pick out curves if you want to pick out the most resistant or the most sensitive cell lines from the plot. And finally, I started off talking about biomarker discovery. And in this case, we also include information about the correlations between individual molecular features and drug response. So here I'm showing an example of what would happen if you search for lapatinib correlating with expression of ERBB2, contextualized in just breast cancer cell lines. And what you see is a forest plot across all the studies that had RNA sequencing information for gene expression. And two of these studies actually have a very strong correlation of 0.7, which is significant after multiple testing correction. So to summarize about PharmacoDB, it integrates the 10 largest pharmacogenomic studies with unified identifiers and unified pipelines. We hope that it'll allow researchers to quickly discover and download the data that's available, and especially if you're interested in particular drugs or cell lines. And if you're a computational biologist that likes to do everything through command line, all the data is also available through a companion R package. It's called PharmacoGX. It's on Bioconductor. So our vision is that PharmacoDB forms the sort of vitrine that you discover data from. You can plan your study, and then you can do your heavy analysis moving into R or a similar programming language. And so finally, now that we've collected all this data, what are some next steps and how are we using it in our own research? So as we've been collecting this data, we identified 7 independent studies that had both drug response and gene expression information. This was over 11 different tissue types in common between them, with 70 drugs that had been profiled in at least 3 studies. And so this gave us 22 million drug response, tissue, gene expression triplets that we could evaluate in a meta-analysis approach. And so our next step is to do this statistical meta-analysis for all of these 22 million triplets. And as you can imagine, very suggestively, I was showing a forest plot that's already in PharmacoDB. So once this analysis is done, this will be available all through our web app. This was supposed to be on bioRxiv for the conference. Unfortunately, it didn't quite make it out in time. However, as one of our preliminary results, we find that you actually, if you do this biomarker discovery in any one specific study, up anywhere from 40 to 60% of the significant hits you'll find are not going to be significant after meta-analysis. And so in my eyes, that really justifies the need to look across independent studies, looking at the same question to really come up with robust results. So with that, I need to thank everyone that participated in this project from both the labs that I'm a part of, our collaborators at our HPC core, as well as at Genentech, all of our funding agencies. The paper is available through the QR code, and the website is pharmacodb.ca. And yeah, thank you for your attention, and I'll take any questions.
Thank you for the talk and the time. We have time for some questions. Go ahead.
Hey, I think it's a great work. So my question concerning whether you have been trying to incorporate the patient-derived samples in your database, because currently there are growing more amounts of the patient-derived drug sensitivity data. So whether that would be also part of the database.
Yeah, so that's an excellent question, and I think cancer cell lines are great because they're easy to work with, and there's a lot of public data, but they're often pretty bad models of cancer. And patient-derived samples are definitely, I think, the future. We're planning to either incorporate this into PharmacoDB or develop a similar database and application for that. One of the sort of differences between patient-derived samples and cell lines is patient-derived sample usually exists only in a particular laboratory. So no one has their usually favorite ex vivo sample used in drug screening. So we have to move away from this idea that you can search for a cell line and rather search for all the, let's say, KRAS mutated samples in the database. And that's something we're working on right now and implementing.
Sure, thank you.
Great talk. I had a related question, so I didn't really get whether toxicity is part of the metadata or feature table for the compounds.
So no, we don't currently have metadata about toxicity for the compounds. I guess, especially where I think that's a good piece of metadata that there are other resources available for that we should probably link to. Yeah. So it's a case of mapping across databases for us.
Yeah, I think it's important. Of course, people screen something that will not likely kill people very fast, but but in many cases it will, right? So yeah.
Yeah.
Thanks.
But yeah, that's a great comment or question.
I got it. Thank you for your talk. And yeah, at least me personally, I appreciate your work here. Especially I heard that you unify the IDs. I don't know whether I missed that, but do you also unify the response of the output, the response metric, especially when you combine different data sets? I guess not all use the same metrics. So some are IC50, some are AUCs, and some probably other ones. I know you have some tool there, but can you comment on the limitations and what do you expect?
Yeah, so I think, what we do is we go back to at least the viability value at each concentration, and we refit a dose response curve using a 3-parameter model, and then recalculate IC50s and AUCs. Not everyone will necessarily agree with how many parameters your model should have or the best way to fit those models. So when you're using our R package, we'll also provide all the published data by the original study authors. But yes, and increasingly the data is available at the well level, so we can also standardize how you normalize to get to the viability numbers, but we haven't done that yet.
Thank you.
Yeah, good talk, and congratulations for the good work. I'm a user of PharmacoDB, and it's a great tool for us. We often find that depending on there is quite a lot of discrepancy in the drug response data, depending on the source from where you get the data originally. Do you have any insights of why that is and what to do about it?
I think in terms of insights, I think there's been some really good work from Peter Sorger's group at, I believe, Harvard Medical School with the LINKS project, where they try to identify technical issues for why you'd see variation. It can often be attributed to the assay used to measure the viability. A lot of the data sets use cell titer glow, which measures ATP content, but not all of them standardized on that. And also growth rates of cell lines might be very different. Unfortunately, a lot of these studies were published before it was realized that it's important to normalize for growth rate in these data. So the existing data won't always have recorded how quickly the cell lines grew. But if there is a resource online, I believe it's called GRmetrics, if you have the growth rate data for your own data, it'll help you normalize and adjust for that effect. And it's actually from our collaborator at Genentech, Mark Hafner. But yeah, in terms of how I deal with this, my approach has been meta-analysis. So especially if sort of my motivation is if I want to translate these biomarkers then into a living patient, well, it's an even more complex system. So I want to find something that's robust to the variation between studies before I can even hope for it to be robust to making the jump all the way back to a patient. So yeah, hopefully meta-analysis is the future.
Okay, there's no more people, but I have a quick question, perhaps. We are on the time. I understand that you have information on sequence variants, or you have?
For
the targets of the drugs.
For which particular variants they are targeting?
No, sequence. I mean, if you have the cell line and you have the resistance, but
yes,
the effect of variants on resistance is quite important.
Yes, so we have some limited information, particularly the CCLE project at the Broad and the GDSC project at the Sanger. They've sequenced their cell lines using whole genome or whole exome approaches. But actually, surprisingly, to me at least, people don't want to invest in sequencing cell lines as much as they want to invest in sequencing patients.
Oh, yeah.
Yeah.
If you were treating because the question will be obvious.
Yeah, so we don't have it for everything.
Yeah, but we do many things on cell lines in the lab without knowing what is there.
Yes, and especially they can be very different between two different labs.
And they're very different from patients.
Yep.
If there's no more talks, I just like to thank the three speakers. Very nice talks.